So we are learning about protein synthesis. And protein synthesis is the main idea or the central dogma of cells. How is a brain cell, and a bone cell, a bone cell? How is a frog cell different from a human cell? It all has to do with the DNA and the sequence of the nitrogen bases in the DNA. That's what gives us our code. But remember, the DNA stays in the nucleus, but the proteins are made at the ribosome. So we see this big idea of how we can take the information from DNA and use that information to build a protein. Yesterday you took notes on transcription, which is the rewriting of DNA code into mRNA. Taking the sequence of DNA nitrogen bases, because DNA can't leave the nucleus, and base pairing, adding new nucleotides to build an mRNA. And so yesterday we took notes on how to do this, and we practiced actually doing some base pairing. And so today we want to take notes on the actual building of the protein. Translation is when we actually build the protein, and we're going to put by joining amino acids. But we can't just put amino acids in any old way. They have to be in order. And so translation, if you look at this picture, translation is where we use the mRNA code to attract these tRNAs. The tRNAs are going to bring amino acids, and that's how we put the amino acids together. It's called translation because we are going from the language of nucleotides. Now remember, DNA and RNA are both made of nucleotides. So we started with DNA and we transcribed it or rewrote it into RNA, still nucleotides. But now we're going from the language of nucleotides to the language of amino acids. So you can see why the term translation actually makes sense. We're going from one language, nucleotides, to another language, amino acids. Now, the place where we build a protein is the ribosome. We want to write in this word organelle. A ribosome is an organelle. It's a small cell part. You can see the ribosome here. This is the cell part called the ribosome. And it will actually join the amino acids together. But the only way to know which amino acids we want to use is to have the mRNA code. So you do want to know what translation is. It's the joining of amino acids. Where? At the ribosome. When? After transcription. And why? To build a protein. But we can't forget the who. We have the mRNA, the tRNA, and those important amino acids. So the steps. The first thing is that the mRNA enters the ribosome. So here's the mRNA. It's always a single strand. The mRNA will enter the ribosome. And when mRNA enters the ribosome, it's going to enter at a specific place called the start codon. We'll see these codons in a second. But I want to remind you that a codon is 3 nitrogen bases, like the letters, on mRNA. So a codon is every 3 nitrogen bases on the mRNA. The codon will actually attract an anticodon. So tRNA is carrying the amino acid. Check this tRNA out. This one's empty, but this one has an amino acid. So the tRNA is carrying an amino acid, and the tRNA will bond with the codon on the mRNA. The tRNA anticodon. Notice this T in the anticodon. It can go with the T in tRNA. So the anticodon is also 3 nitrogen bases. But it's 3 nitrogen bases on the tRNA. The tRNA is almost always drawn kind of looking like a fork, where this would be the anticodon. Something like UAG. Now, if I see U, I know that it's RNA. So remember, whether it's tRNA or mRNA or rRNA, there's never any thymine. We would only see uracils. Now, this anticodon, UAG, it's an anticodon because it's on the tRNA. This anticodon would only be attracted if the mRNA was the right base pairs. Now remember, we're talking about RNA, so I have to use the RNA base pairs. The correct RNA codon would be AUC. AUC would be the mRNA codon. That AUC attracts the UAG, and at the top of this tRNA is the amino acid. Now, as this picture shows, this only happens when the ribosome is there to help this occur. So these are the 3 main steps. The mRNA arrives, the tRNA comes in and bonds, bringing the amino acids. Once we have multiple amino acids, they will begin to bond together. So you can see this bond being formed here. When we create a amino acid chain, we actually use peptide bonds. Peptide bonds. And these are strong bonds. They're what hold the amino acids together. We've mentioned before hydrogen bonds. Hydrogen bonds we always call weak. Peptide bonds are strong, and they hold together the amino acids. Amino acids are held together by peptide bonds. We'll keep going. So every codon, every 3 letters, codes for 1 amino acid. AUG gives me methionine. GCC gives me alanine. And we'll keep going until we reach what's called a stop codon. A stop codon means that we are done. We'll release the amino acid chain, and then the amino acid chain will fold into a specific shape. And that's how we build a protein. That's when the protein is complete. And so those are the important steps of building a protein. But now we actually want to practice what that looks like. Because we use a codon chart to find the correct amino acid. Here, it's telling us what it is. But what if I ask you to find AGC? AGC. What amino acid would we get from AGC? So let's write that out there, AGC. But before we do that, I want to see if you can work backwards. So for example, AGC is one of these amino acids. Is it threonine? Is that what AGC is? Well, threonine is next to the letter A. It's next to the letter C. But it's next to the letter C. This threonine is actually ACC. Notice first letters are always here along the left side. So threonine does go with the first letter A, but it goes with the second letter C, and we wanted the second letter G. So this means that I needed to be in the A, but over here where the G comes down, and it meets up with that C. And that, serine, is AGC. So what would this one be? It says the word stop. It's one of our stop codons. Is it AUA? No, because we have to start with the first letter. So stop is actually A. What about Lucy? This one. What is that one? Can you find the correct codon or mRNA for Lucy? And then, what if I give you one like GAU? Can you find what GAU equals? So as you finish, you're going to do your summary box. But I'm also going to ask that you find the correct code for Lucy, this Lucy, and that you find me what GAU is. That's the end of the notes for today.